So now I'm going to open the series Back to Basics with confocal analysis. So sorry in advance for those who are not using a confocal microscope. And for the other, I hope it would be helpful for you. I wanted to share how I process my data. I know we are not dealing with the same materials, or we are not facing the same issue, but I think that maybe some elements or some methodology can be reusable with more or less modifications. And so just really quickly, but the advantage of the confocal microscope compared to other wide field microscope is that you can obtain images of fluorescence emitted by your sample at the level of the focal plane of the objective, because the aperture, so the pinhole, is arranged to block out all the blurred information and to only detect focused information. So the good point is that you have really good quality images. But in addition, and that we are going to discuss today, is that it is possible to perform hyperspectral or maybe multispectral imaging. So thanks to the presence of a bunch of lasers containing different sources of excitation. And at EMPMC, this is the sources of excitation that you have. And thanks to the presence of 32 sensors, which allow the detection of the fluorescence signal over an emission range from 420 nanometers to 722 nanometers, and with a step of 10 nanometers. So basically, one channel is 10 nanometer. And all the data I'm going to show you were acquired with that set of parameters. And so, of course, there is many ways to process your confocal data, but I'm going to show you with Fiji software, because first of all, it is an open source software, and it has this great advantage to be compatible with that files. And besides, there is a large community, so there is that contribute with a lot of plugins. So sometimes the hardest is to find the right plugin. And I will use the Stowers plugin. So Stowers from the Stowers Medical Institute. And I will show you how I pre-treat my data, how I perform a blind linear unmixing analysis, and how I extract the fluorescence spectrum from selected pixels or from a region of interest. So I am doing on live. So let's say that I open one of my sample. Okay, so you can check the metadata. And here, so you have the 32 plus 1 channel of transmitted light with different fluorescence. And the third thing I am doing is that I duplicate the data, and I always work on duplicate data. So I close it. And so, okay. So, and then I delete the last channel because it is a transmitted light channel. So image, stack, and delete slice, and current channel. Okay, so also, but it is optional. It will depend on your acquisition parameters. but I am deleted because, you know, I am using 3 different excitation sources. And so I am also using bandpass filters, which reject the light coming from the lasers. But after you have a spectra like that, so if you want to avoid that, you have to delete the channel corresponding to the reflection light, I would say. So for me, it's this channel, but it will depend on your acquisition parameters. So I'm not going to do that because I have already done. But this is my raw data corrected. And so now I can work on it. So the first thing that you can do, it's like an exploratory work. And you can use a very nice tool, which is the dynamic spectrum. So coming from Stowers plugin again. And so you have a spectrum that is changing when you move in. So you can see here the spectrum. So
Juliette, sorry, I just sorry to interrupt you. One thing I wanted to make sure: why did you have to delete? What did you have to delete exactly before? I understood well what you did with the data, but what is that you have to delete some? What is exactly to be deleted actually before you process your data?
Deleted the channel.
Yeah.
This, yeah.
And
yeah.
What is the problem with these channels you have to delete?
Because they are okay. So if
they correspond to excitation lines.
Yeah, excitation.
Okay, that's to filter out the laser lines. Okay,
basically,
okay.
This is the first filter, the first
perfect.
light. Okay, and so you see it's black and so on. So I delete them.
Perfect.
And they are suppressed. The excitation lines are suppressed on the microscope by filters.
Yeah.
And then you get, that's the reason why you get those dark images in there.
Yeah, exactly. Okay. But yeah, perfect.
Okay, so dynamic spectrum. So here you see the background. Here you see some signature. And here you see that I have a different signature, for instance. Okay. So my issue was that I have a sample, so it's stromatolites from Sardinia, and of course there is many fluorescence signature, but of course unknown for me, and I would like to separate spectrally the different component. Okay, and don't forget that we are working with hypermap. So my hypermap, 1 pixel is 1 spectrum. So if you see here, we have like around 2000 times 2000 pixels. So around 4 to 5 million spectra. So this is a lot. And the question is really how to explain the whole fluorescence signal with a limited number of spectra. And to do that, we are going to perform principal component analysis. So we are not using PCA, we are using a different algorithm, but which is close to PCA. It's non-negative matrix factorization. And then it's allowed to have the eigenvectors and to have the spatial distribution of your fluorescence signal, so to have the eigen images. And thus to do that, you have to go to Fiji, NMF. So again, from Stowers plugin. So you can start with 2 species. For instance, here I think I have at least 2 species. For the background, I would say 0. I am working with 8-bit images, so the saturation level, the max is 256. the number of iterations. So of course, the higher the better, but it costs analysis time. So you have to deal with that. What is really great with that tool is that if you have an idea of the spectrum you are expected, you can check the option initialize from plot. And so you can fix one or whatever spectrum you want. And if you have the spectrum, the reference spectrum, you can download it, but I don't have any. So I cancel. And which is great also is that you can have the residuals. So I'm not going to launch the calculations because it will take like maybe 3, even 5 minutes, maybe more long, depending on the power of your computer. But so here are the results. So open. So for 2 compounds, I obtain these eigenvectors. And I obtain these eigen images. So, okay. So first of all, maybe for the eigenvectors, what I like to do is that extract the data list, okay? And then I plot it in another software, so Excel or Origin, whatever you want. And don't forget to convert the number of channels into wavelengths. And if you deleted some channel, don't forget to delete the wavelengths also. So like that. So come back to our process. And so now for the eigen images, so I like so you see I've got 2 different fluorescence spatial distribution. So I like the fire representation because you can see the more intense pixel. But you can change the color, image, look at table, and you can choose whatever you want, spectrum or whatever. What I like to do also is to make a composite. So to do that, I have to choose first one unique color for each channel. So let's say that the first channel is green because it is emitted in green. So I choose green. Here, I used to use red because it is emitted in red wavelengths. But don't forget that some people cannot distinguish between red and green, so I use magenta. I like to enhance contrast, for example. So here, and okay, I apply it. But if you enhance contrast, it's better to have the same settings for all the channels. So you press set and then you press propagate to the other channel of the image. Okay. And then I do my composite, so color and make composite. I can also set the scale. Okay. So I know the parameters, but okay, global. And finally, I apply my scale bar. So, okay, a bit bigger. And you can choose to put the text or not, whatever. Okay, and finally, I convert the stack into, is color and stack to RGB. And you have your composite. Okay. So go back to my sample. I did.
Juliette, what was the second image then? So the first one is a composite, is the overlay, and the second one was.
Sorry, which image?
On your former, on the one that was open, there were 2 buffers, right? Or 2, no?
And mixed this one?
Yeah. Oh no. Okay, it's the same. Okay, there's only one. I thought there was
yes.
there were
no,
2. The bar in the bottom was okay.
No, it's the same. It's the same.
And Juliette,
Yeah.
the eigen images,
Yeah.
they are characterized by okay, so they come out of the PCA-like analysis, right?
Yeah.
But they are characterized then by they are not characterized by one spectrum, right? But they are characterized by a range of spectra which are close to each other. How does it work with the spectra of the eigen images? In the eigen images, because you still have some contrast, right? So the spectra are different.
Yeah. So dynamic spectrum. What is that? Sorry. Field spectrum. So here you have the first eigenvector.
Okay.
And if I put the first channel.
Yeah.
Okay. So if okay, so this is particular with my sample because I don't know if I have pure component. I mean, even this eigenvector is maybe already a mix of different components. But with labeled, I don't know, cells or, it works perfectly because it's really characteristic of a fluorescence signature.
Okay, but this on the left, it's the eigenimage corresponding to the
first eigenvector.
Yeah. And in this image, what is the reason for the contrast? It's intensity differences. It's what is getting different colors?
Okay. Yeah. So I'm going to come with that.
Oh, but, okay, sorry.
But my assumption is that in the most intense pixel, I have almost the pure component.
Okay.
And the contrast is that you have less pure component with that kind of spectra.
Okay. Thank you.
But it's my assumption.
Excuse me, Juliette, I have another question.
Yeah.
The eigenvectors are linear combination of
for this
spectra.
algorithm.
Yeah, okay. And how did you choose the number of eigenvectors? For example, according to eigenvalues, do you have the eigenvalues that explain the variability of the?
No, the fact is, okay, usually what I do, it's really exploratory, but what I did, I checked the residual. And so I'm looking if there is some, did I have some?
The residual is the difference between the eigenvector and each values?
For instance, for instance, as I have my residual here for 2 compounds. And you see, so normally what its residual is in white. It was for 2 compounds, so this white zone remains. So I say to myself that maybe there is another eigenvalues, but I don't have. I know that you are expected. I don't have the weight of each eigenvectors. Is that you are expected? The weight percent of the contribution of each eigenvectors?
Yeah. So you have to try some.
Yeah, I don't have them.
Some combination. Okay.
Yeah. Okay. I know that.
Sorry, Juliette. So when, for example, you use you take like 2 or 3 eigenvalues, does it give you at some point any weight of those different?
No, I don't have the weight of each eigenvalues.
Okay.
The only thing that I could do is to compare the eigenimages. And for instance, here I have two components, here 3 components. And after I said to myself, maybe the second component is not relevant enough because it may be part of the first component. But the other thing I'm doing is that I want to extract the fluorescence spectrum and to compare them. And that I'm going to show you after. Is that okay?
Yes.
Okay. So the third part is so to extract my fluorescence spectrum, because here, as I don't know really, because it's natural fluorescence, and with NNMF algorithm, it is supposed to be like fluorescence signature, but of course, and what you say, all that it's close to reality, but not necessarily the reality. So I need to have the emission the fluorescence signature. And so to do that, so I'm going to use that, close and close. I have my raw data and my unmixed. So first of all, I have to demix my. Okay, so I have a stack. It was the previous eigen images. So color and stack and stack to images. So I have my first compound, my second compound. And I have to create a binary mask from my first compound. So using the tool, the threshold. And I'm not going to use the automatic mode. I am going to use a very high threshold because I want to select the more intense pixel, because I'm making the assumption that I have the pure component in the most intense pixel. So this is an arbitrary value, but I am choosing to select just the pixel, the 0.1% of this pixel, of this image, and then apply, and then I have my binary mask. And then I want to apply this mask on my stack image. So I use this imaging tool, which is mask stack, and also from the World Institute. So the stack is my raw corrected data, and my mask is my unmixed compound 1. Okay, and so I get a masked images. So it is a stack, but with only the selected pixel. And from that, I create a spectrum. Okay, so create spectrum. So for the statistics, you can choose sum, average, whatever. I choose sum. And thus I have emission spectrum, which is more close, which is closer from the reality. Okay, and also I can extract my data list and plot it in another software. And okay, I would like to show you with the second compound, because here you see that it's going to be more easier. Because obviously we have a chlorophyll signal. So with automatic mode, it is working. You know, it is thresholding the right object, but apply the same threshold as previously, just to have the same condition. Apply mask. Okay. Run and mix 2. And I get my masked images, and you see I have my, and I create the spectrum and the same. And you see, we have a chlorophyll signature, which is more realistic than the eigenvectors, even if the eigenvectors was close. And so that's it. So here I did it with 3 compounds. And what I noticed is that for my first and my second compound, the spectrum seems to be similar, but the green spectrum is lower intensity, but it seems to have the same shape. So maybe it's not relevant to pick three components. By comparison, I performed this analysis on microbial light from Mexico, so with 2 and 3 compounds. And I think that maybe the third component was not relevant. And finally, when I extract the fluorescence spectrum, I found that I think that my black spectrum is different from my green spectrum. So I think that I have 3 different spectrum, and I think that I would keep 3 components to explain my fluorescence signal. So that's it. I showed you all the steps I knew for processing my confocal data. And I hope it was clear, and you can ask questions about it, and we can discuss, of course. And I will share with you all the protocols so you can use it as many times as you want.
Thank you, Juliette. That was really great. That was a nice one. I don't know, so might be Chinese for people who have never used confocal, but that was super clear. And personally, I'm happy with it. So is there any questions, comment?
I have a question / comment, I suppose. Juliette, we have already talked about this for like specifically for cyanobacteria when we looked at the data from the confocal. I just wanted to clarify. So like towards the end, when you do the full, like the sum spectra of the fluorescence across the different slices, right? And you see like how many components you see, and they kind of are they matching your eigenvalues, right? Which you use in your principal component analysis.
Yeah.
But is my understanding correct? Like if you just have, let's say, bacterial cells where the signal is chlorophyll,
yeah,
then if you just do a sum signal, and you first of all see in your sum signal that the majority of your fluorescence signal is just chlorophyll, then you have the answer, like it's just one component. That's that right? Okay.
Yeah. If you know that you will have only one fluorescent signal, you can do what we saw together. You just have to extract your fluorescence spectrum from your images.
Right.
Yeah. Okay. So the third part, I would say that you apply a binary mask and then you create your fluorescence spectrum.
Okay. Yeah. But if we do that, and if I do see like there is an additional component from that signal, then I have to go to the eigenvectors and see and do the process. Okay.
And in addition, I really think that if you are dealing with bacteria, I mean with biological stuff, I think that eigenvectors would be really close from your emission spectrum.
Yeah. No, okay.
Because basically, it was this plugin was developed for labeled dye imaging.
Yeah. Okay. It was super clear. Well, thank you so much.
But for cyanobacteria, so they are autofluorescent, and when it looks like when they are doing well, you might see, I mean, depending on the species or the strains, but for some of the strains that we are looking at, the ones that are doing well, they might fluoresce in you see mostly chlorophyll, so they will be bright red. Now, when they are not doing well, they might fluoresce in the green. So you would have 2 components, and it seems that there might be also transient things like red and green, and more red or more green, and so on. So that's when deconvoluting like that might be interesting.
Yeah. Yeah.
I don't know if you, maybe François, sorry.
Yes.
Oh, no, it was just very detailed, but when you get at the end, you get the image with your components. It was not clear to me when you show the components. Of course, you have a fluorescence spectrum for each component. But is it possible, or maybe it's what is made, I don't know. Is it possible to stack all the pixels, all the spectra, and to get a less noisy spectrum from, I mean, you see what I mean, right? To stack all the spectra to get the average spectrum of this component, but multiplied by the number of pixels where it is. Which would maybe it's what is done, but because the spectra of each component, they looked a little bit, you know,
you mean?
yeah, it's not a critique.
But this is what Juliette does with the ROI.
Yeah, but so it corresponds to how many spectra, how many pixels, or you stack all the pixels of the image and you get the spectrum of that.
Yeah, I stack all the selected pixel, pixel with threshold, and then I have the average, not the average, the sum of the spectra of the sum, but all the selected pixel. But of course, you can choose a more selected pixel. It was because I did the spectra on a few pixels because for my case I assume that the more intense pixel was maybe pure components.
Are they pure? Otherwise, they are polluted by something else. Okay.
But if you have only pure component in your sample, it will be easy.
Yeah. Okay. Thank you.
And could you get the superimposition of all the spectra that are represented by a given eigenvector? Is it possible in order to see the diversity in a given class?
I don't. You mean this?
If I clearly understand, you can get one eigenvector that is a linear combination of a lot of individual spectra.
Yeah.
So is it possible to superimpose all of these spectra and together with the eigenvector in order to see the diversity and the variability of the spectra of all of the pixels that are represented by a given eigenvector?
I'm not qualified enough to answer that, but I even don't understand everything.
One eigenvector represents a lot of pixels.
Yeah.
It's a class of pixels.
Yeah.
And for each of these pixels, you have an individual spectrum.
Yeah.
So I wonder whether there is a lot of diversity of all of these spectra that are represented by a given eigenvector.
Okay. So of course, with this plugin, it is not possible because it is an imaging plugin. So it is not statistic, not developed for statistic code. Maybe it would be able to code it or to add some statistical clarification, but with that, no. It's not,
but maybe the residual value answer this question. It's the diversity between each spectrum and the eigenvector.
Yeah.
Maybe it's the Euclidean distance between each spectrum and the eigenvector.
Yeah, I remember that we tried to do something with the withdrawal and to convert the pixel, the remaining pixel, yeah, the remaining the square of the remaining pixel, and to see if it was diminishing with the increase of the spaces. That's it.
I guess the residual is probably the Euclidean distance between the spectrum and on a given pixel and the linear combination of the different eigenvectors.
Yeah, so you can get the diversity and the variability within a given class of pixel.
Okay. Okay.
Yeah.
Maybe we could discuss together of that to do something more stronger.